The purpose of this video is to review lab information dealing with the blood. We will be observing slides representing the different types of blood cells. The pointer on this first slide is on a red blood cell, which is called an erythrocyte. It is shaped like a biconcave disc, and it contains the red pigment hemoglobin, which is used for transporting oxygen. It has a nucleus when it is first formed, but it does not have a nucleus as a mature cell. The red blood cells you are observing here do not have a nucleus. The next group of slides represent the white blood cells. There are two groups of white blood cells, the granulocytes and the agranulocytes. The granulocytes have granules in their cytoplasm, and they have a lobed nucleus. There are three types of granulocytes: the neutrophils, the eosinophils, and the basophils. You will notice that all three names have the same ending. The first granulocyte we will observe is the neutrophil. It is the large cell in the center of the slide. It has several lobes in the nucleus, and it is the most abundant white blood cell. The second granulocyte we will observe is the eosinophil. Its nucleus is bilobed, and it is easy to recognize because its cytoplasm stains red. The third granulocyte is called the basophil. It is the least common of all the white blood cells. We will not observe this white blood cell on the slide, and you are not responsible for identifying this on a test. The second group of white blood cells are the agranulocytes. The agranulocyte does not have granules in its cytoplasm, and its nucleus is not lobed. There are two agranulocytes, the lymphocyte and the monocyte. You will notice that both names have the same ending. The first agranulocyte we will observe is the lymphocyte. Its nucleus covers most of the cell. The second agranulocyte we will observe is the monocyte. It is the largest white blood cell, and it is easy to identify because it has a kidney-shaped nucleus. This is a review of human blood types. Human red blood cells have antigens, also called agglutinogens, on their cell membranes. There are two antigens, antigen A and antigen B. The ABO blood typing is based on these antigens. Blood type A has only antigen A on its cell membrane, and blood type B has only antigen B on its cell membrane. Blood type AB has both antigen A and antigen B on its cell membrane, and blood type O has neither antigen on its cell membrane. The human blood plasma contains antibodies, also called agglutinins. There are two antibodies, anti-A antibody and anti-B antibody, that are associated with the ABO blood grouping. These antibodies develop in the plasma spontaneously about 2 to 8 months following birth. The anti-A antibody fights against antigen A, so it will not be present in type A blood, but it will be present in type B blood. The anti-B antibody fights against antigen B, so it will not be present in type B blood, but it will be present in type A blood. Blood type O contains both anti-A and anti-B antibodies, and type AB does not contain either antibody. In review, looking at the chart, you will see blood type A, and it has only antigen A and its antibody is anti-B. And then if you look at type B blood, you'll see that it has antigen B, and it has anti-A antibody. And then blood type AB has both antigen A and antigen B, and it has neither anti-A nor anti-B antibodies. And then finally, type O has neither antigen A nor antigen B, but it contains both anti-A and anti-B antibodies in its plasma. This is a review of blood transfusions. If the wrong antigen and antibody come in contact, this will cause the red blood cells to clump. This process is called agglutination. A person does not have to worry about his own antigens and antibodies reacting, but there is a danger of clumping if a person receives the wrong blood type in a transfusion. It is always best to use donor blood of the same type as the recipient blood. In an emergency, it may be necessary to use another type of blood other than that of the recipient. The two main factors to consider are the donor's antigens and the recipient's antibodies. The blood being donated may also contain antibodies, but there are not as likely to be as many antibodies in 1 or 2 pints of blood as there would be in all the blood of the recipient. Because type AB has no antibodies, in an emergency, AB could receive blood from any other blood type. Therefore, it is called the universal recipient. Because type O has no antigens, in an emergency, it could be given to any other blood type. So it is called the universal donor. In reviewing the chart, you will see that type A blood should receive only type A, but in an emergency, it is permissible to give type O blood. O is the universal donor, and it does not contain any antigens. Type B should receive only type B blood, but it is permissible in an emergency to give type O. Then type AB blood should receive only type AB. But in an emergency, because AB is the universal recipient, it can receive a transfusion of A or B or O. And then type O should receive only a transfusion of type O. For blood transfusions, you must also consider the RH factor. Each blood type can be either RH positive or RH negative. For example, if you have O positive blood, you should receive a transfusion of only O positive blood. If you are AB negative, then you should receive a transfusion of only AB negative blood. This is a review of blood typing procedure. If you were typing your blood, you would need an anti-A serum and an anti-B serum. You need 2 drops of your blood. Place anti-A serum on 1 drop of blood and place anti-B serum on the other drop of blood. If only anti-A serum clumps your blood, you have type A blood. If only anti-B serum clumps your blood, you have type B blood. If both anti-A and anti-B serum clump your blood, you have type AB blood. And if neither anti-A nor anti-B clump your blood, you have type O blood.